Welcome to this video on cell signaling and receptors. We will explore the mechanisms of ligands, receptors, and the important nuclear superfamily pathway. Cells communicate through secretory signals categorized by their range. Endocrine signals travel through the bloodstream. Paracrine signals target nearby cells, and autocrine signals act on the secreting cell itself. Receptors are divided into three main types. Cell surface receptors receive hydrophilic signals. Intracellular receptors wait for lipophilic signals inside the cell. Orphan receptors are similar in structure but have unknown activating ligands. Core signaling relies on two components, ligands and receptors. Ligands act as chemical messengers, while receptors are the proteins that change shape upon binding, triggering a signal within the cell. Intracellular receptors reside inside the cell. To activate them, small lipophilic ligands must pass through the plasma membrane. Examples include steroid hormones, thyroid hormone, retinoids, and vitamin D. The nuclear receptor superfamily is a conserved family of transcription factors. They regulate gene expression and response to those small lipophilic signaling molecules. Nuclear receptors have a modular structure. This includes the N-terminal activation region, the DNA binding domain, a flexible hinge region, and the ligand binding domain, which facilitates dimerization. Receptors dimerize in two ways. Homodimers consist of two identical units, often associated with heat shock proteins until activated. Heterodimers consist of two different units, often already bound to DNA in the nucleus. In the steroid receptor pathway, a hydrophobic ligand diffuses into the cell and binds the receptor. This causes the heat shock protein to dissociate, allowing the receptor to dimerize, enter the nucleus, and bind to specific DNA sequences.